I'll just get started. So, I mean, as John said, my name is Michael Cunliffe. I work both at the MBA and the University of Plymouth. And Maya's asked me to give a coffee time talk today on marine microbiomes. So one of the things that Maya wanted me to do was to really, I guess, make this talk quite inclusive and introductory. And so I think it's quite important before we talk about marine microbiomes that we actually really define what a microbiome is. So the human microbiome is something that, if you're not a microbiologist, might be more familiar to you. It's in the social media, it's in the popular press. Lots of people are familiar with the concept of a healthy microbiome in the guts, for example. And so a common way to think about the microbiome is the diversity of microorganisms, so the microbiota. So this is things like bacteria and archaea, but also eukaryotes like fungi and protists and viruses. So often we see this as being described as the microbiome, but what's actually critically important when we're thinking about microbiomes is function. So it isn't just the list of microbes that we find in our guts, for example. What is critical is the function and the activities that they have. And so microbial function, microbial activity is a very broad and very complex topic, which I don't have the time to go through here, but just a few sort of concepts for you to think about. What matters is, what are those microbes made up of? What are the structural components? So, what proteins are they made of? What lipids do they have? One of the really critical things that microbes do is they make stuff. They make metabolites, they make enzymes. These can have positive impacts, so lots of the gut microbes that we have make compounds that are useful for our healthy working guts. But also microbes make things that are dangerous, for want of a better turn of phrase, that are toxic. So this is important. Another thing to really get clear, which isn't so obvious if you're not really used to working with microbes, is microbes have activities that are internal, so cellular activities, things that happen in the cell. But microbes make a tremendous amount of compounds and molecules that they secrete into the external environment, like toxins and like enzymes. And so this all needs to be taken together when we think about the microbiome. So when we say microbiome, we mean microbial diversity, but we also mean function. And again, it's obvious to say this, but I think it's quite important just to have it clear in our minds when we're talking about microbiomes. We need to put them in some sort of spatial context. So our microbiome needs to be in a biome, it needs to be in a spatially defined area. So in the case of the human gut microbiome, the gut is the biome, and the microbiome are the microbes in there and the functional activities that they. For those of you that are interested in microbiomes generally, or if it's a concept that you're not super familiar with, this is a really nice review that's just come out conveniently in the journal, rather conveniently also called Microbiome. And this is an open access review. You can download it freely. It's really nice, and I think it covers the topic quite well. So we have the human microbiome, and of course we're interested in marine microbiome. So these concepts that I've just said, microbial diversity, microbial function, going together in some sort of biome, they apply to the guts of human beings. They also apply to the marine environment. And one of the really interesting things about the marine environment, but also one of the really challenging things about the marine environment, is when we're thinking about the biomes that microbiomes are in, scale is really important. So in my research group at the MBA, one of the biomes that we're interested in is the Atlantic Ocean. So we think about what is the microbiome of the Atlantic Ocean. This is a Google Earth image here. You can see it's principally the sort of the North Atlantic. We study the microbes in this system across a very large spatial scale. I mean, the Atlantic Ocean is the second largest ocean basin on the planet. Pretty huge. Marine microbiomes go from this sort of scale down to this sort of scale, to microbial scale. This is a fluorescence microscopy image of a single diatom cell. It's a large centric diatom, costodiscus, and it's a fluorescent image. So what you're looking at, those little red coffee bean structures inside the cell, they're the chloroplasts of the diatom that naturally fluorescent. And then the turquoise structures, so the turquoise structure in the center, this is a fluorescent dye that stains DNA, so we're looking at the nucleus. And then all those specks on the outside are actually individual bacteria living on the outside of this diatom. So we can look at this diatom in terms of its microbiome. The biome is the diatom, and it's got microbes living on it, and they have a functional activity. And this, I guess, illustrates the beauty and complexity of marine microbiomes. If you think about the Atlantic Ocean, that has a microbiome, and then every diatom that's living in the Atlantic Ocean has its own microbiome. Complex and quite fascinating, really. So in my research group at the MBA, we're really heavily focused at the moment on trying to understand microbiomes in the open ocean, particularly in the deep open ocean. To the left of this slide is an infographic, not from me, it's from a recent publication in Nature, really trying to highlight how poorly studied this environment is. I include this infographic because for those of you that attended David Sims's Coffee Time talk relatively recently, he also showed the same infographic. And David works on big sharks that feed on the cephalopods that we get in the deep open ocean. In my research group at the MBA, we work right at the other end of the spectrum. And you can see in this infographic aggregates. So aggregates are particulate organic carbon in the open ocean. They are critically important for being the base of the marine food web in these systems, but they also have a big role in transferring carbon from surface waters to deeper water. I've included this slide, just especially for those of you not familiar with doing marine microbiology, just to illustrate some of the challenges that we have. So one of the places that we study the deep open ocean microbiology is at an observatory called the Porcupine Abyssal Plain Sustained Observatory, which is maintained by NOC, and it's just out into the Northeast Atlantic. And we've been on research cruises to this site and we've been sampling. So I think it's important to get a sense of scale when we're thinking about these systems. I mean, this infographic is quite cute, I guess. It illustrates some key points, but it doesn't give you a sense of the scale of the system. So when people from my group have gone to the Porcupine Abyssal Plain to do work, they've gone on this research vessel, which is the Discovery. Discovery is almost 100 meters long, so it's actually 99.7 meters long for those of you like detail. So if we actually took Discovery to the PAP site, PAP is so deep, it's almost 5,000 meters deep. It would take 50 Discoveries stacked end to end to get to the bottom of PAP. How do we sample the microbes in this mesopelagic, bathypelagic zone of PAP? Most of the sampling that we do is done on this sort of water sampler. So this is basically a rosette with some water bottles on the side. You can see it more clearly in this image here. This is held over the side by a crane, and it is literally winched all the way down to the bottom of the seafloor, nearly 5,000 meters, and then water samples are collected and then they're brought back up onto the ship. And then when they're brought onto the ship, you can see they're being collected here, they're taken into a lab and then processed. So we do this type of work in my lab. We went on a cruise last year and we bring the samples in and we process them, and it's a big activity that we're working on. But there's other ways to study marine microbes and the marine microbiome. And so one of them is to actually use other people's data sets that they are willing to share. And this is just another example from work that we've done in my group. So the Tara Ocean expedition is a principally French-led circumglobal survey of the marine microbiome. This is the ship that they did most of the work in. This is the Tara Ocean yacht. They basically travel around the world, sampling the ocean, collecting seawater, and using mainly molecular tools to study the microbiome. One of the, I guess, really core principles of how people are doing this work now is that people are willing to share their data. So when the Tara scientists have finished analysing the data for what they're looking for, they make that data publicly available, and then other people can use it for their own interests. And so we've been using these data sets, particularly looking at fungi in the open ocean and trying to understand what functional roles that they have. So this is a figure from a recent paper from one of my postdocs, Nathan Christmas, that just came out a couple of months ago. And you can see each of these dots is a sample, effectively, is a station where samples were collected from the Tara expedition. And what you can see here that's quite striking is the sampling bias that occurs in the open ocean. So lots of samples collected from surface water, slightly fewer samples collected from the DCM. The DCM is the deep chlorophyll maxima, and then much fewer samples are collected from the mesopelagic. And this really illustrates an issue that we have, certainly in trying to understand the marine microbiome, is there is a bias towards surface water and much less well known is about the mesopelagic. So we analyse these samples, and we're particularly interested in fungal, a group of enzymes that fungi have called glycoside hydrolases. So the previous slide I said to you that we're interested in aggregates and particles in the deep sea. These are the enzymes that fungi would have to degrade those particles. So we want to know which enzymes that they have, where do we find those enzymes, is there any indication of what the functional roles of fungi might be. It's a complex figure. I'm not going to talk about it in too much detail. Check out the paper if you want to know more. But just a summary, if we actually look at these enzymes in surface waters and in the DCM, you can see most of the dominant type of enzyme is a cellulase. So these are glycoside hydrolases that degrade cellulose. Cellulose is one of, if not the, most abundant polysaccharide on the planet. It's in plants, it's also in a lot of green algae as well, so that sort of makes sense. Maybe fungi are processing algal biomass in the surface waters. But what's quite interesting when we look in the deeper mesopelagic, and again it's important just to reiterate, there's a skew in the sampling bias in the mesopelagic. There's much less sampling done down there. We see a greater range, a greater diversity of these types of enzymes. And again, I'm not going to talk about in too much detail, but we see other types of enzymes becoming, I guess, more relatively abundant. So these are things like glucosidases, which are used to degrade glucans. So glucans like laminarin, for example, from seaweeds, or chrysolaminarin from diatoms, is an exceptionally abundant polysaccharide in the marine environment. Also chitinases as well. So chitinases degrade chitin. Chitin, well, hopefully most of you know that chitin is a really abundant biopolymer. It makes the exoskeletons of invertebrates like this copepod here. And chitin is also an interesting polysaccharide in that most of the other polysaccharides are very carbon rich, whereas chitin is carbon rich, but it also contains lots of nitrogen as well. So it could have more value for the ecosystem because it contains both carbon and nitrogen. So we do this type of work. We either go out on research cruises ourselves and we sample the microbiome, or we utilize open source data. And we also work in the lab with model organisms. And I guess the point I want to make with the transition from the last slide, which is global ocean surveys of microbiomes, to this slide, is the importance of scale. And so we look at fungi in the open ocean across, we compare the Indian Ocean with the Atlantic Ocean, but we also study these organisms at microbial scale. So what you've got here is a chytrid. It's a type of fungus, and it's actually growing on a chitin bead. So we're particularly interested in chitin for the reasons that I just said. So this image on the left, this is a light micrograph of one of these chitin beads. So this big sphere is a bead of chitin. It's about 100 microns across. And then these two sort of spheres with these swirly, swiggly things are chytrids. So these are the fungi. So this is the cell body, this sphere, and then this is this structure. This is called a rhizoid, and this is how the chytrid attaches to its substrate and how it feeds on its substrate. So the image in the middle is it's the same thing. It's a chitin bead with chytrids growing on it, but this is just the electron micrograph. So it's a much more high-powered microscope image of effectively this. And you can see here the chytrid. So this is the chytrid sphere, the cell body. And then we've got this rhizoid structure coming down, attaching to the chitin bead that's sort of all around. And you can basically see in this image very clearly what the chytrid is doing to this chitin bead. You can see these structures, these furrows that the rhizoid has created. There's a big one here where this thicker part of the rhizoid was. And basically what this chytrid is doing is dissolving the chitin. It's producing extracellular enzymes that are dissolving the chitin away, and it's feeding on the chitin as a substrate. So these are two microscope images, a light microscope image and then a confocal microscope image. And then what we actually do in my group is we actually take these microscope images and actually create models using those images. So this video now is a model from a real chytrid growing on a real particle. So the chitin particle is in beige and then the chytrid is in blue and green. So hopefully this video plays for you. So what we can actually see here is the rhizoid in blue, which isn't in contact with the particle, is really basic and spread out and diverse. But the rhizoid in green, which is becoming clearer now, the part of the rhizoid that's interacting with the bead, you can see this is much more structured, more branches, more tips, and this is part of the adaptation these fungi have to deal with particles, where they essentially increase the surface area contact with the particle. This is the work from David Slowden. He's a PhD student in my group. He's just had a paper published in Proceedings of the Royal Society B for those of you that are interested. So we're interested in microbes in the ocean, we're interested in microbes in the lab, and we're interested in what functional roles that they have. And this is this essence of the marine microbiome. Another big area of interest in my group now is trying to take those themes of studying microbial diversity and studying microbial function, and to do this in the Arctic. And I'm just going to play this video in the background. So this is a video from NASA, and it shows the state of ice in the Arctic and how this has changed since the early 1980s. So the video will run. It starts in January 1984, and then it ends, I think, at the end of 2019. And what you basically... Oh, 1 second. Okay, so the video started playing. So, two things. What's been happening in the Arctic? Well, we've seen a general, on average, trend in the reduction of sea ice. So we get generally less sea ice each year, and particularly in the summer, there is less sea ice. And models predict that the amount of sea ice in the summer will reduce so greatly that it's potentially the Arctic will be ice-free in the summer. And what's also important for the Arctic is the type of ice that's present is changing. So in this video, this lighter coloured ice is multi-year ice, so up to 4 years old. So most of the ice in the Arctic is going to be what we call single-year ice, so ice that only formed from the previous winter, and we are losing some of this multi-year ice. I'm not going to talk about this so much more because this is work that's in progress right now. We've had a lot of activity in the Arctic over the last few years. So we've been working with collaborators at Newcastle on these big Russian rivers that flow into the Arctic. We took part in 2 cruises last year in the Fram Strait and the Bering Sea during the summer to try and understand this system. We've been collaborators with the Mosaic expedition, which is led by RV in Germany, where the ice ship Polar Stern basically followed the ice for a year through this system. And then a cruise that we should have gone on this year, but was actually postponed because of COVID-19, but it will happen next year. And it's, I think the one that I'm most excited about is on the Swedish icebreaker Odin. And the icebreaker next summer will actually take this track here that you can see, this yellow track. And it will actually go through the different types of ice, so multi-year ice and single-year ice, and we'll study this system. If there's any younger people in the audience, I just want to highlight a recent publication that we've published. I'll just play this video again. As researchers, we tend to publish papers in scientific journals. Those scientific journals then get peer-reviewed by other scientists. This is a paper led by Berta, who's a postdoc in my group with Rowena Stern at the MBA, and I and others are co-authors. So we basically wrote a research paper on our work in the Arctic. This is one of the figures from the paper. But quite importantly, this journal, this Frontiers in Young Minds journal, is actually peer-reviewed by school children. And so if there are any younger people in the audience or anybody who's got any kids and they want to know a bit more about what we've been doing in the Arctic, this is an open access journal. It's freely available, and I'd be quite keen to get sort of the perspective and point of view, hence the sort of maybe simplistic figure here. So I just want to make some acknowledgements. We're pretty well funded from a range of funders, and that's great. This is my research group at the MBA. I've mainly talked today about Nathan Christmas's work, so he's a postdoc, Davis Loudon's PhD work, and some of the work that Berta Zanker's been doing. So Berta's doing most of the work in the Arctic in the group, for the group. And finally, I've been asked why this is a coffee time talk to include this slide by Maya. I guess if you're, well, you should all be members, but if you're not a member, it's, joining the MBA and becoming part of the association is really important. There's lots of benefits to being a member, and you really join this international community of marine biologists. So I can stop there.
Thank you, Michael. Excellent stuff. Took us through nicely. I will stop the recording now and go to any questions.